Hi, welcome to another episode of Dose of Pharma. Today's episode centers around inspiring alumni, where we get to know an alumnus who started in the same position with the same tools that we have doing the pharmaceutical science course at Monash. We explore how they utilized and took advantage of these tools to seize opportunity and to create an impact on the development of our course. Once again, I'm Jess, and today I'm joined by Christine, who'll be my co-host for this episode. The inspiring alumni that will be our guest for this episode is Carson Newhan. He completed a double degree in engineering and pharmaceutical science, completing his final PharmSci year in 2016. He's a very curious person, which is demonstrated through his passion for data analysis. This is exemplified how, when he was given a poster assignment to explore HPLC, he went above and beyond and created a virtual HPLC chromatogram database that is still used in the unit today.
So thank you, Carson, for joining us today. Let's jump right in and start off with, I guess, an intro question. Yeah. So although I guess a few years ago, we're just wondering which side of your degree did you enjoy more, the chem eng side or the PharmSci side?
Yep. I think it's kind of interesting question. I think in my third year, I was like, yes, I love the pharmaceutical science part because it's like so different from chem eng, and it's like much more involved, a lot of lab work, much more practical. And then the engineering that we learn is like heavy assignments and also a lot of like theoretical question and a lot of hard calculation. So I would say like back in third year, I definitely enjoyed the pharmaceutical science more. But then as we continue in our engineering component, I start to find that engineering is actually quite interesting in a way, although you still get like the stress from the assignment and then the tension like with teamwork and everything. But in fact, like engineering, actually we get to learn a lot more like high level or harder like concept, and we got more time to practice like math and physics, which is like subject that I actually enjoy. So so at the end of the day, I would say like I enjoy both component, although in terms of like the time I found like myself develop the most is definitely like during my third year with pharmaceutical science.
And part of like the PharmSci is the HPLC, which we're going to talk a bit about today.
Yes.
Yeah, for the listeners who don't really know what HPLC is, so that's like high performance liquid chromatography, which is like an analytical method used to identify, separate, and quantify compounds. And that's like really important in research and industry. So then as part of that unit, there's a simulation program that you developed back in 2017, and we still use it in this course as part of that. So that's pretty cool.
Yeah, to bring back some memory. Still remember like back in those days when we doing this unit, we have to sit in the lab like from 9 o'clock in the morning, and then the lab lasts for like more than 3 hours.
Yeah.
And if you fail like your morning run, you might be able to stay back to do your afternoon run. So you end up leaving like 6 o'clock.
What inspired you to create the HPLC model, and like how did you go about doing it?
I think that's like definitely like the idea of like making it more efficient and making the process faster. I think that's like part of it, like why like the HPLC simulator will help. Originally, I was just thinking in the lab when running the HPLC, we only like investigating like discrete or individual like condition. And I always wonder what will happen in between if we change the solvent mobile phase percentage to 50 to 60. What happened to 55? And and just like those are like a little bit of question that I ask myself. I love like Laurence like giving us this chance to explore more about HPLC at the end of the unit. So we actually given permission to enter the lab and then do more experiment and then build this big model that like big poster that including all the different parameter that will change the HPLC, the output, and how they're going to change it. So that was like the original idea of the whole HPLC model. We just want a poster that introduced the parameter and then the output and how they interact with each other. When you get a little bit deeper into it, you realize that, it's not quite straightforward, especially in another unit that Laurent teach. It's about tablet making one, I think.
Yeah.
When we start to talk about design of experiment and then interaction between different things, like different parameter and input, I just like making the connection. I was like, what if we can just make a HPLC simulator that take into account all these input and turn it into like a chromatogram that we normally see. If you just click it, we don't have to wait for like the 3 hour that we do in the lab. And apart from that, we can also use this as a prediction compared to real life experiment. And I found that, that would be cool to do. So that's how it started, I think.
No, as Laurence would say, that's awesome.
Yep. I think she did say that. So, yeah.
Yeah, that's really cool. And I guess, as you said, there was a lot of like long days involved to get all that data. I'm not sure if you can really quantify this, but how much data was required to build this kind of program? I guess, did it take months, weeks? Well, I guess, how much data?
That's a really good question. I think the formal number of ingestion that we did and the actual data that we included during my simulation is about more than 100, 150 ingestion, I think. But then ingestion as in each sample point. Some of them take like more than 3 hours, some of them like quick, like 20 minutes. But then that's just the good data. Well, some of them are not that good, but the good part. But then there's a lot of like other background works that we have to do to investigate. If like something's go wrong, we might have to redo the run again. I would say like at the end of the day, the data that we are using is about more than like 200 ingestion. That's not including the data that we obtain in the class. Another thing with the unit is that it generates a lot of data because when students run in the lab, like they will obtain a chromatogram that get passed on to the next year, etc. So in our year, we actually get to have some of the information from the people who did the unit before. And then in our year, we also generate some data. So including those, I think, we probably have like more than 400 different data points. Yeah.
What resources did you have to help you with that? Was it just like the previous year and like everybody in the class helping or?
Oh, okay. In terms of resources, I think I actually rely quite a lot. What's the website called again? Chromatograms, I think. That's where I learned all the basic theory, and then I got a little bit deeper since the website it has a lot of interactive activity or interactive e-learning things that we managed to do. At the same time, it doesn't give you enough background information regarding chromatography and the mathematical model, etc. So at the end, I actually also found some resource from the book. I can't really remember the author of the books. But it's like one of those books like that have a lot of simulation information on how to predict the outcome. It doesn't include simulation, but it does have like quite a few like formula and everything. Yeah, so that's like basically the knowledge side, like in terms of resource, because it's like something that I don't think like anyone in pharmacy campus did, like in the Pavil campus. I don't think anyone has actually like really dig deeper into like how chromatogram, how HPLC work, and what's the model behind it. So I think a lot of it is just like me trying to like figure out like what's going on at the background and learning all this.
You're really driven by your own curiosity.
Pretty much, I would say. Yeah.
That's really interesting. You mentioned that you had all this data that there's only, I guess, 150 good ones. So I guess there was a lot of runs that you did that weren't successful. I know for me, I'm currently running HPLC for my honours project, and there are setbacks every single time. I guess what were some of the challenges or setbacks that you did face with the project, maybe with the machine, or I guess collating all the data together?
Yeah, this is like one of the story. When we collecting the data, we have this trouble with the pH. Since that's our first time like actually preparing a pH buffer run on HPLC. Like in the lab, normally we weren't allowed to do that because, well, not like we weren't allowed, we just avoid doing that because it can cause precipitation in the pump and it can clog the machine. So yeah, so when we actually doing that, it was like, oh no, we actually don't even know what we're doing. So we have to look up a lot of information and we actually consult different people, like how to prepare the buffer for HPLC and how to make sure that it doesn't precipitate in the pump, etc. At one point, I think we did end up precipitate some of the buffer solution in the pump and causing like a clogging. So that was actually pretty stressful. It's
it sounds it.
We're thinking like, are we breaking this $1 million machine?
Oh gosh. Yeah, it's already stressful enough when you like, do something wrong in a normal lab where like you're not doing something with some very expensive equipment. I can't imagine how stressful that would have been with a HPLC machine.
Yep, definitely. Oh, well, I mean, yeah, it's also like almost like the end of the day, like getting close to 6 o'clock, and we were not allowed to be in the lab after that. So yeah, I think like at that point, communication become really important. Yeah, we just need to let Laurence know what's happening and what could potentially be the issue. While we're trying to resolve it, we're trying to figure out like exactly what's happening. So to figure out like there's a clogging issue because of the bed pressure is rising and it's not pumping properly. So yeah, that's like a little bit troubleshooting on the spot, and then figure out the issue. We have to talk to Laurence to figure out something like what we're going to do next. And luckily, I think someone from Shimazu come and managed to clean the pump, and it works. So that was good. At the end, like I don't think there's like any major damage to the machine itself. Yeah, and we're actually able to obtain like some valuable data as well. So that was good. And yeah.
Thank goodness.
Yep.
Yeah, you mentioned to solve that problem, you had to really use your communication skills. And you also mentioned previously that that's something you use a lot in your current job. Did you learn like any other skills from this experience of creating the simulation?
Oh, yes, I think, yeah, a lot. Part of the project, because I was like kind of leading or in charge, managing like the people who actually doing the ingestion, a little bit of a communication between like different group. I have to like basically keep up to figure out like a lot of micromanagement to figure out how they're doing and everyone's what stage they are at. At the same time, also giving them a lot of support. Like if they have an issue, with the machine, if they have a problem, they always like welcome to talk to me. That's like the part basically just trying to maintain my presence so that if you have an issue, you can always come to me. I think that's like a big part when managing a project. Yeah.
Yeah, that's really strong leadership skills.
Yeah. And the other part of the communication skill that I really pick up basically is the second stage of the project. Initially, I developed a program on MATLAB. Basically, it's a engineering software that we used in engineering. But then it has its limitation. It's not widely available to everyone. So in the second stage of the project, we're trying to move everything online. And then that opened up a completely new door for me since I never programmed anything, any website before. And then we have this collaborator actually from, I think, United States. Yeah, they're from NCU, I think, University of Northern California. Or yeah,
that sounds right.
Yep. Yeah. So basically, they know all the programming, and then they know how to build the website. But then they have little idea about the actual program and the mathematical model that used for the HPLC. So I think like a big part of my job is actually communicating what the model is like and how can we implement that into the frameworks of the website. And that's like meetings and visual meeting as well, and to work through different parts. And then I also learned a lot about programming from them.
Yeah, that's so interesting. I guess like how was the experience of stepping up to become like the project leader? I guess during undergrad you're usually in teams for group projects, but I guess there's never really one lead at the very top of those group projects. So how did you find that experience?
Yeah, it's actually it's pretty new to me, I guess, back in those days. It's actually my first time actually managing such a big project. We divided into a small group, and then each group there's 4 to 5 people, and then I was also overlooking the whole operation. It's like a lot of responsibility, I guess, because you have to make sure that everyone is doing the right thing and everyone is following the SOP, etc. At the same time, it's interesting, I would say, and encouraging, because with all the problem that we faced, I was like, we can do this together. It's not just my problem. It's like everyone can help in a way. So even though I was like at a higher up, like I just feel like, I'm kind of like one of them. And because we all studied the same course, we're able to, relate to each other in many ways. So at the end of the day, even though I was the leader or the management, like the manager role in this project, I feel like I just like working with a lot of my colleagues or like my friend, that we're able to achieve this together. And I think like just building the morale is like one of the important things, and that's why like this project is like one of the one that I really enjoy, I guess.
Yeah, those skills definitely would have helped with like all the other group projects we have to do at uni, because we definitely get given a lot of group projects.
Yeah, indeed.
And definitely to be able to like step up, but then still be like, okay, we're kind of like on the same level still.
Yeah, I think like sometimes like things can happen, and it's like, if you're like one of those much higher level, and then people won't be willing to approach you, and then sometimes that could actually be a stumbling block for actually hearing like what other people has, and sometimes they might have way better idea than what you have as well. And that's something that I found out when I was managing this project.
And like making sure people are comfortable to come to you with their ideas and stuff like that as well.
I think that's like a really important skill for a leader.
Do you think, I guess, your experience leading that HPLC project, do you think those skills have helped when, I guess, you've gone out into work field now and you're working in the industry and working with others? Do you think those skills, I guess, have been translated into the work field?
Yes, I think definitely. Well, let alone like all the technical skills that I have, programming and also like operating HPLC. Those are still with me, and sometimes I still need to use some of the programming skill that I use in other projects. So that has been transferable, like pretty much. But then I think like more importantly is the soft skill. Since like that's my first time actually stepping up to taking on the project, working in a team. And I think like that really contribute to later in my course and also currently in my role. I'm actually able to speak up when things happen, actually make an important decision for the team and also for the other project that I was working on. Especially like another important skill is like time management back in those days. With university, when I was like doing the project, there's a lot of other assignment that happen at the same time. And just like managing time.
Yeah, that's like I'm really having to get familiar with at the moment, because this year I've got a few subjects where rather than exams, I've got a like semester-long assignments, and I'm finding that really different, like to deal with, rather than just like not having to work on stuff throughout semester, then cramming before the exam pretty much. But to have like more of the sense of balance between everything is definitely like an important skill to gain.
Yeah, definitely. Yeah, and something like with the engineering course as well. At the end of the year, we have to do this like big project that is, it's like a semester long, and you don't have any exam, but then you just have like a lot of individual submission and then the group submission as well. And it's like, it's pretty intense, and you have to really work together as a team. Definitely the experience that I have with the HPLC project really helped me in my final year design project.
Yeah, I guess that's interesting that you have, I guess, touched on engineering, because although it's a different degree and course, there's a lot of aspects that are shared across both of those courses. So maybe what aspects do you think of engineering were you able to integrate with the pharma side degree, or vice versa, so you were doing both of them for a while?
I think like for pharmaceutical science, Laurence really touched on the idea of critical thinking. That's actually a really huge part in engineering, because engineering, you have to make a lot of decisions, and then you have to think outside the box sometimes. in order to prevent bad things, especially in terms of safety. So that's like a part that I felt like I'm really benefit from doing the double degree course, something that I really learned from PharmSci. Apart from that, in terms of how engineering contribute to PharmSci, engineering actually get touched on a lot of hardcore math and engineering concept that PharmSci just skims through or not even like mention. So I think just being able to integrate both, especially during the development of the HPLC model, engineering actually give me like much more understanding how exactly the full flow inside the HPLC machine. Well, the PharmSci actually give me like the practical perspective, like what are some like equipment and each component and how they actually impact on the chromatogram at the end. So just combining those two and actually drawing the link between the things that I learned throughout both engineering and pharmaceutical science. I kind of like enjoying that process, just making the link. And
to understand things more in depth.
Yeah.
You definitely seem like a very curious person who would want that.
Yes. It's funny though, like since sometimes in engineering there's actually overlap with pharmaceutical science. Say we have this colloid chemistry unit that we actually did in engineering, and yeah, and you actually like see, well, although it's like similar content, that's the same colloid chemistry, but you see it in different perspective. Like one of them is like, well, what's the theory and the equation and how this equation come up? That's like engineering. But in pharmaceutical science, it's like, oh yeah, now we have this equation. How are we going to formulate something? Maybe using this equation, or maybe just use this equation to test the final result. Or we can just like formulate something to fit other perspective or other specification. So yeah, so that's like pretty cool. I think it's just seeing like both the practical and the theoretical side combining together. And that's like one part that I really enjoy the double degree. Yeah.
To conclude things, just what would your advice be for students who want to be innovative and to seize different opportunities like you did?
Oh, that's a very good question. In fact, for me, I think be curious, be open to all these different idea. And when you do have innovative idea, try to hold on to it and be resilient. Since like one thing that with the HPLC model, the simulator that we developed is in fact much more like this time. There's a lot of times that I was thinking, I don't think I'm going to finish this. There's too much stuff like happening with life and with uni, and then yeah, this looks pretty daunting. And also I have to learn a completely new programming language to just fit the model in, and there's like so many bugs at the start, and it's like, oh, I don't know if I can finish that. But at the end, like yeah, just breaking the things into chunk and being resilient, and don't be frightened by the amount of the work you're going to do. I think that's like an important I don't know, alright, the skill is actually more like
mindset or
mindset, yeah, something along that line. Attribute,
yeah,
quality, yeah. Yeah, I think yeah, just don't be afraid of what you're doing. Since I can't say for sure, like if you're starting something new, there will be a lot of works and it's stressful. So sometimes just hold on to that, your idea, and actually take ownership of it. And yeah, and try to be resilient and pursuing what you're doing. And that's how you actually be able to achieve, I think, success. Yeah.
Yeah, that sounded like really good advice just then. Because it does get very overwhelming and it's very easy to be afraid of all the work and all the stuff, especially when starting with something new. But yeah, to take control of it, yeah, it seems very important.
Yeah, definitely. I think that's like, actually not only just apply to like university project or anything, it also apply to life in a way, I guess. Yeah.
Thank you for letting us talk to you. It's been a very interesting conversation.
Yeah. Same here. Thanks for this chance, like this invite to be able to be here in the Vodcast. And yeah, it's pretty cool to be the guest here tonight. Yeah.
Yeah, thanks so much again. Same, I learned a lot through talking to you, so it was good.
Yep. Yeah, hope you two will be doing well, and can't wait until to see how you actually make this project one of the interesting one and something that you can talk about in future.
At least we don't need like 400 data sets for this.
Yes. Well, I mean, what you're doing is something cooler, I think. Yeah. Very different field, but at the same time, I think it's really relevant.
To conclude the episode, we wish to acknowledge the people of the Kulin nations on whose land we are gathered today. We pay our respects to the elders past, present, and emerging. For those listening who may be based elsewhere, we pay our respects to the traditional owners of the land from wherever you may be listening from. We especially welcome any Aboriginal or Torres Strait Islander people listening in today.